Hello friends, welcome to lecture 2 part 1 of the week 4. So in this lecture we will look at the basic concepts in genomic and transcriptomic analysis before we proceed for a new genome, and in the next part we will look at some case studies with examples of how we perform the genome analysis. So the first question is which genome is to be sequenced? What should be the criteria? For example, if you see here, there is a genome of peacock is sequenced, right? So the criteria in this case was because it's a national bird of India. It is very beautiful bird, right? The hypothesis is that from the genome we can get clues for its phenotype, that why it looks so beautiful, and the long feathers and beautiful colors and feathers, right? So basically this was the idea. So when you are approaching this, you also have to look at that whether it is sequenced earlier or not, right? If and what has been sequenced from its genus or family, right? So that will help in basically doing the comparative analysis. So all these things are very important while you go for genome sequencing. And also, like if you see in this slide that we show that some of the plant families that have been sequenced. So you can see that some families have been sequenced in detail, whereas some families are lagging behind. So basically why it is so, it will always be like that, because first of all those sequences which are of high priority because of their commercial value or environmental value, ecological value, right, or because of national interest, so those, or maybe because they're getting extinct or endangered species, so these will be always be of higher interest, and that is why their families will be sequenced more. And this will also affect when you go for the comparative analysis, because you will not find many genomes which are like yours, but that is also an advantage because you are doing something very new, or when you are doing something which for which several other genomes from the same species or genus or family are already available, then basically it will add up some more analysis part in the when you do the comparative analysis. Okay, so basically what is needed for a genome sequencing project? First of all, you should have a clear objective and significance of the genome, that what you want to sequence, why, because earth is full of so many distinct species, right, and in almost every corner of the earth, and it could be a eukaryotic genome, it could be a bacterial genome. So deciding that what you are going to sequence is, and what is your hypothesis, that why you want to sequence and what are you going to get out of it, right, is very important. Okay, because whatever you do, you will spend almost equal amount of time. Now it will be like whether you choose a good genome for sequencing initially, or you pick something which was not so relevant at that point of time. Okay, and then in addition to new genome, sometimes resequencing of a genome is also a question, that you want to resequence because of various reasons, like the previous assembly was not good, right, or it was basically done in detail but it had a lot of errors which you found, or you think that in your region the species is different, okay, and that comparative analysis is important for health or disease or ecological perspective, right. Then what you are going to sequence, you have to confirm. So confirmation is very important, because morphologically they may look similar, okay, but when you do the molecular level confirmation, you may find differences. And also it is the first of the steps in genome sequencing project, that you should confirm your species by molecular methods using some standard molecular markers. Okay, then another question is what tissue sample to be used. So basically when you are doing a animal or plant genome, the idea is not to harm the animal and do the sequencing without basically any invasive method. So generally, like you have to take ethical approvals also before proceeding for any such genome, right. So then you have to take the sample which is convenient to take. For example, it could be a scraping from the skin, okay, or it could be like blood, but you cannot draw blood, okay, like for example tiger, you cannot like sedate a tiger and draw the blood at any point of time. So generally, like the sample collecting person or who is giving the sample to you it could be a zoological park or national park, right, where those animals or birds are present. So they will also give it only when those animals come for treatment or their routine health checkup. Okay, so that time you can get these kind of samples. Otherwise basically some tissue for which you don't have to cause any damage or harm to the animal are one of the easiest methods. Okay, and then you have to extract the DNA and RNA for various molecular biology and sequencing experiments, right? So the choice is like either you use a kit which is already available, so which has also advantages and sometimes a good choice, but sometimes the tissue is difficult to get the pure DNA or high quality DNA out of it. So then you have to modify it or go for a conventional method where you will optimize the method first and then go for the final DNA extraction, which is which you will be using later for the sequencing. Okay, so at this point the quality of DNA and RNA is also equally important because that will determine the quality of your sequencing and ultimately the assembly. So, and also you should avoid the contamination by other organisms. For example, you are extracting a tissue and there could be protozoa or fungi present. So basically your this sequence that you have got is also from the tissue of interest, that is bird or animal, but also from the contaminants which were symbionts or present on their that tissue part like protozoa or fungi. Okay, similarly bacterial contamination could also be there, and you may end up sequencing more of those genomes and less of your interested genome. So that is important. Okay, then basically when you have decided all these things, that what genome and confirm the genome, and okay, so basically you have to see that design the sequencing strategy. Okay, like how much coverage is sufficient for the genome, that how much should I sequence so that I can get a good quality genome. Okay, so there are some formulas and methods to determine this, which we will be doing later, but in this I will give some clues about it. Okay, so that is very important. And then in that estimation you also have to look at how much should be the long read and how much should be the short read, because ultimately you will be going for the hybrid assembly approach, and this will the balance between the two, or the appropriate quantity of the two is very important, because that will decide that what assembly statistics you will reach at the end. And if your assembly statistics are not good enough, you may not go for chromosomal or high C sequencing. Okay, then based on the genome size you have to see that okay, do you have sufficient computational system available with you, or you are taking help from someone, right? Then after that you have to design appropriate phylogenetic analysis, okay, for constructing a phylogenetic tree, so that you can show that how different or distant it is from the other closely related or distant genomes. Okay, and which species can be used for the comparison, and there should be reasons for comparison, that why you have included only those, because you cannot include each and everything for comparison. You cannot compare a bird with an elephant. So basically you will be considering species which are closer to that organism, okay, and also take an outgroup. So there are some standard methods of phylogenetics which we will also see later. Okay, right, and basically we want to identify that how that species evolved, and for which you have to see the signs of adaptation. So there are like multiple signs of evolutionary adaptation that you may determine from the genome, okay, by the comparative analysis, and those will give clues of what set of genes or pathways or basically things in that genome have evolved, so that the genome looks, the species looks so different or basically fit in that environment, right? And then you have to interpret the results, and there should be a hypothesis with which you started, and you may test that. Okay, it could be true, or you may find something more for to support that hypothesis, or it may fail. And in addition you should keep an open mind, because there could be new observations, okay, and you should interpret them in an unbiased manner, and then go for the final basically interpretation of the results. Okay, so there should always be a scope for new findings, because the genome that you've considered, like if it is how new that is, that will also determine that what are new findings. And also basically, like it also depends upon your knowledge level. You should read the literature around it, and that will give clues to you that what question you should try to answer from that genome. Okay, then the data, once you are ready with all this analysis and findings and your results are ready, then you should try to submit that data to a public repository such as NCBI, because that accession number will be needed when you submit a manuscript. And you should compile all the introduction methods, results, and discussion that what you got from that genome, okay, in form of manuscript, and communicate to a appropriate journal. Okay, so basically from a genome you can do genomics, that is determining the genome of that organism, including the genome assembly, annotation, okay, genome analysis, phylogenomics, okay. And at the same time you should also do the transcriptome, okay, where you are determining that what genes were expressed, what are the alternate spliced products from those genes, right, what was their expression level, okay. And this will also give empirical evidence when you are doing the assembly of the genome. So transcriptomics data is also used for the genomic assembly, because that gives like true evidences that the genes that you have predicted or found from the genome were actually present, right. Then proteomics is another dimension to this, that you want to determine that basically there were transcripts, but you do not know how many of those were expressed as the final protein product, and right, and their respective proportions. So this is important also to find or get a quantitative estimate of also the pathways that are present and their expression levels, and okay. And these days, like metabolomics is becoming important, because this will tell that you might have found a particular pathway or the set of genes which are enzymes which are converting subset A to product, okay. Now finding that product, which are the final products of any molecular pathway or this enzymatic biotransformations, okay, or which are a result of overall metabolism of a genome, you can determine that using metabolomic approach, where the mass spectrophotometry such as GC, LC, or MALDI will, or basically NMR will help you. Okay, so the challenge in genome sequencing is that genomes are very large, okay, like bacteria could be in millions base pairs, okay, less than like 10 million base pair, and the eukaryotes could be in 500 million to 2, 3, or even like 16 billion, for example the aloe vera genome. So they could be very large in size, okay. And most of the sequencing technologies can do only short reads, or even the longer reads that do like PacBio or the nanopore ONT. They are not sufficient enough to complete the entire genome in one go, okay, like this or like this, okay. So basically what you need here is a combination of the two, and basically a computational assembly and scaffolding, and basically reconstructing the genome is still a challenge, okay. So basically these are some of the challenges that are still there because of the technological limitations, but things are improving, and now long reads are making it very much possible to construct the entire genome from head to tail, okay. And then when you are doing a genome, there are many things to be considered. For example, you have to determine what are the coding regions, that is the proteins. So in a genome, typically only 2% of the genome is coding, which actually translate for proteins, and about 25 to 27% is genic, which has both genes and the coding part and the non-coding part, that is the introns, okay. Then there are intergenic regions, okay. So you have to truly call and identify the genes which are coding, and also in the terms of their introns and exon structure, right. There are a lot of non-coding sequences which could be regulatory in nature. It could be expressed transcripts, or it could be the genomic sequences present in the intergenic regions in the chromosomes. that includes like tandem repeats or transposable elements, simple and complex repeats, right? And retroposons, and these are all complex repeats. Okay, so basically if you see in this picture, there are all these things you have to determine when you are doing a new genome, and these data you have to report that what is the respective composition of the genome and the kind of contribution from each of these parts. Okay, for example, in case of human genome, we now know that like 25% is genic and 75% is intergenic, and in this basically there will be like coding region and the intron constituted by introns or pseudogenes or genic non-coding region. Okay, then repeats occupy a lot of part. About 20% of genome is unique, which is non-repetitive, and we do not yet know the function, right? And this like tandem and interspersed repeat, like simple and complex repeats. So basically now we know these percentages, okay, which are actually new for a new genome, and when you do a new genome you have to report these percentages or this kind of map for that genome. Okay, so this is one of the important outcomes of any genome sequencing analysis, right? And other challenges include that sometimes the genomes contain high amount of repeats, such as in case of humans it is like 50 to 55%. In case of plants it could be like as much as like 70 or even more. Okay, and they may also show high amount of heterozygosity, which may complicate your assembly and reduce the assembly statistics, right? And polyploidy, like how many copies of the DNA is present, like human genome is diploid, 2 copies, similarly diploid, tetraploid, and these things are common in case of plants. Okay, so that will be a challenge when you do the sequencing strategy, that how much you have to sequence, and also it will be difficult to resolve later. You have to do more analysis to see the whole genome duplications and impact of those duplicating genes on the genome, right? So repeats, heterozygosity, polyploidy are some important aspects when you do the genome sequencing. Okay, typically like heterozygosity, for example, k-mers, when you are doing the when you have sequenced the genome and you have got the reads from which you derive the k-mers, and those are used to see that how many peaks you are getting. So like this is a typical low heterozygosity genome, but when the heterozygosity is more, you will get like 2 peaks. Okay, so this part we will also cover in detail later, but basically these kind of analysis gives clues that basically what is the level of heterozygosity for your genome, and it is high or low. More than 1% is like kind of high, right? And also the literature may also tell that what if somebody has done like a preliminary analysis or reported the heterozygosity for that genome, so that you should always be good with the literature and read more about the genome of your interest, okay, which you are handling. Okay, so because that from there also you can get some clues, and then you can validate using the data in hand. Okay, so like this is a simple chart of the repetitive sequences, and like heterozygosity, determining the content of all these repeats is very important, for which there is a program called repeat masker, which needs some training data, which is also a task, right, which you have to do, right? And then basically it will divide the the genome or classify the genomes into these kind of repeats, including their respective proportion, and the table that comes out of it, okay, also goes as a main or supplementary table. And the interpretation of these repeats is important when you are handling that genome. Okay, so basically it all starts with a cell that is there in the tissue that you have considered. Okay, you will be extracting the nucleus, and from the nucleus basically you may or may not extract the nucleus, but basically one of the ways is you take the nucleus out, find the chromosome, find the various DNA. Okay, at chromosome level you may not be able to separate. You may try using flow cytometry, but it is still a expert and challenging task. Okay, and then the DNA that you have isolated will be sequenced as reads. Okay, so basically read is part of the okay, your chromosome. Okay, it represents only a small, read means reading. Okay, so I have read this genome from this place to this place. Okay, so this is one read, and it depends upon the sequencer that you have used. So if the sequencer is like 150 base pair, your read length here will be 150 base pair. Okay, so we in previous class we have also done this like these definition, but quickly we will do a review. Like shotgun sequencing, because the sequencing technology can accept only shorter fragments, so you have to break the DNA into smaller pieces such that it is appropriate for the sequencing. So basically breaking the DNA into fragments is important for whole genome sequencing. Okay, that is amounts to shotgun sequencing. Okay, then read is something that you are determining the order of nucleotides and the identity of nucleotides, right? Okay, so read is like reading the DNA. Okay, the DNA is read. Okay, so basically if this was the complete chromosome, so you have read this part of the DNA. Okay, and like it could be like 150 base pair or based on the your genome size. Okay, so read, and then it could be like you are reading the DNA from a single side using okay one primer, or from both the sides. Okay, single end and paired end reads, right? So basically if you see in this picture, you can see that this is the chromosomal DNA, or the DNA that you have isolated. Okay, read is like you are reading this part of it. Okay, so basically the determination of A, T, G, C, A, C, A, G and whatever nucleotides are there, okay, from one direction. But if you are doing from both the directions, so it will be like paired end. Okay, and then basically since you are doing it from a genome and okay, doing in pieces, so again you have to go back to the genome. So you know, you have to do the assembly process. So if there is some nearby genome available, right, then you can do the alignment to that reference genome and basically keep on aligning so that you can reconstruct the entire sequence, right? So it could be a de novo assembly, or it could be a reference based assembly in this case. Okay, so other definition is like when you are assembling the reads. Okay, so basically you try to stitch the reads on the basis of the overlap that they have. Okay, and based on that overlap they will be assembled into contigs, right? And you keep on stitching by looking at, like, this is one read, there is another read like this, so there is some overlap here. So by this I can construct this longer read, and then there was another read which was like here. So okay, so based on this I can even extend this one further. So this way you keep on extending in both the directions this contig. Okay, and once you do not find anything which is at this part or this part, basically you stop and that becomes a contig. Okay, and then basically arranging those contig in a particular order is scaffolding based on some clues from transcriptome or from longer insert sizes, okay, that you considered for sequencing. So those build these scaffolds, right? So contig is one thing. Okay, then assembly, like putting those contigs back into the reference of a genome, right? And then since you are doing it for the first time, there will always be gaps in the genome. Okay, there will always be some pieces missing because you could not cover that by the sequencing. Okay, because there are many places where you can leave the DNA. For example, when you are doing the pipetting, at that point it is not that basically you are dealing with like some micrograms of DNA for sequencing. So that some microgram for library preparation may not truly cover the complete DNA. So you may miss some pieces there, and plus when you are fragmenting that DNA, there will be some pieces which got okay even removed that point of time, DNA fragmentation. Then when you are making the library, it is not that all the pieces will get adapted and other processing steps. So you may lose some DNA there. And then when you are doing the sequencing actually on the flow cell or in an nanopore or packed by or rolling circle, so there also it is not that all the DNA that you have put will be sequenced, right, because of the capacity of that system. right? So that way ultimately you will be leaving some part of the chromosome which remains unsequenced, okay, and that will constitute a draft sequence and not the complete sequence where the identity of each and every base pair is known. Even for the human genome, basically it started in 1995 but finished in 2022, so it took like more than 23 years and to complete it, and that tells the story that to complete a 2.9 billion nucleotides genome, 22.9 GB genome, okay, which was of interest to all over the world labs, okay, still it took like more than 20 years to complete, okay. That means it is a true challenging and difficult project to actually complete it, but draft genomes you can find so many which are available in various public repositories and published papers, okay. Okay, scaffolds is like arrangement of contigs in reference to the genome based on the long distance clues, okay. KB is a unit like kilobases and million bases, okay, which is commonly used to tell that the size of the genome, okay, of that or that piece which you have in hand, okay. Then assembly of read into contig, like if you see that this is a sequence which is, so in the chromosome it is present at this position and at this position, so basically one sequence is present at two positions, so which may complicate the assembly part. So basically one read was generated from this part, one from this part and one from this part randomly. Basically you can not ensure that this read will be generated, but if there are chances that these reads are generated because of random fragmentation and sequencing, okay. So basically you can see that this read covers, okay, this contains this repeat or basically duplicated region but has some unique region, okay, and similarly here, okay, and this way you can basically cover the entire region, right. So assembly of reads is important, the correct assembly, because otherwise these kind of regions which may be present at same identity at multiple locations may complicate the process of assembly, okay. Then basically there could be a de novo assembly where the pieces that you have you are trying to assemble, okay, by overlap between them each other and using the various de Bruijn graph and other strategies, right, and constructing the de novo assembly or a chromosome, or basically you can use a template for a closely related genome or resequencing project of the same genome and try to map your reads against that and stitch them and build the complete assembly. Okay, so hybrid assembly thing we have talked earlier also. So basically we are using the short reads and long reads both to do the assembly, right. Generally like shorter reads will be required in more depth or more quantity as compared to the longer reads, because longer reads itself are already assembled, and to reach to this kind of piece using these short reads, okay, you will require a lot of assembly and these kind of correctness approaches, but basically long reads are that way better because they are longer and they cover a large part of the genome, okay, and that will avoid the assembly issues also, okay. Basically, and there will be always like these kind of reads will be more accurate, this will have more errors, so a combination of the two will help to correct each other, okay, and improve the quality as well as the basically the assurance that the assembly is correct, okay. So like short reads are high throughput, very cheaper, and basically very less error rate, okay, but may be issues because of you've amplified that region or short length, and basically in repeat regions which are few kbs, a 150 base pair read can be aligned at any point in that one one or 2 kb region, and that will create problems. But from the same region, if you have, for example, this is a repeat region and a short read is here, so the exact repeat, this repeat can also be placed here because the regions are same, okay. But if you have a longer read, like this was the region repeat, okay, read, right, but if it is a longer region, then basically this will cover this repeat part as well as the unique part also, okay. So which will that is why the long reads are better. but and short reads can help to correct these the errors in the long reads. Okay, so long reads have higher contiguity, low cost, and quick sample preparation because of their technologies. Okay, and for repeats they are the perfect choice. Right, then basically you can have a complete genome if you have determined the order and identity of each and every nucleotide in that in the all the chromosomes at that genome. Draft version a genome with gaps where some portions still need to be sequenced and not known. Okay, coverage is like how much you have covered that genome or read that, how many times you have read that genome. Okay, for example, there is a typical formula, like there is a hypothetical genome of 2000 base pair. Okay, so basically the formula is n into L divided by G. Okay, so basically if you use it, like there is a hypothetical genome of 2000 base pair of 8 reads with an average length of 500 nucleotides. Okay, so the length is, so basically you can say that number of reads are 8, length of reads are 500. Okay, and genome size is 2000. Okay, so this will be 2x coverage. Okay, so basically you have covered that genome using this number of reads and length 2 times. Okay, so this way you can estimate that how much reads or data you need to cover that genome. Okay, generally like 200 times or more by the short reads, and at least 25 to 30 times or 25 to 30x by the long reads is good for a hybrid assembly. Okay, then this picture tells that the sequence of the human genome that I was talking about, which took almost to determine those 23 chromosomes, took almost 23 years, and from all over the world the efforts were made, and then now we have the complete sequence of the human genome, and these things are so important, and that is why they are covered in prominent journals such as Science. Okay, so this was the first genome that came in 2001. Okay, the first draft, which was of 2.9 billion base pairs and just 5-fold coverage. Okay, using the Sanger reads. Okay, and then basically in 2022 this complete sequence of human genome came, which is after 32 years. Sorry if I mentioned 23 earlier, it was 32 years after 32 years, right, from 1995 to 2022. The first telomere to telomere, that is end to end, from telomere to telomere of the complete chromosome, it was sequenced, right, and again in Science, right. Okay, in addition to the genome, transcriptome is equally important, and it tells, even just transcriptomic analysis is sufficient to make a publication also, because transcriptome tells that what are the genes which are present in that genome, their expression levels, the alternate splice products, and also gives a lot of clues about the pathways which are present. Okay, because it is basically determining all the express part of the genome, which is more functional, although it may not give you clues about the genome structure, duplications and repetitive regions, but at the functional level it is very good and sufficient to tell a good story. Okay, so basically transcriptome is like a set of all the RNA which is present in a cell at that point of time. Okay, and okay, so basically we extract the RNA, convert that into cDNA, and then go for sequencing. Okay, so it tells the genes which are expressed, their expression, regulation, alternate splice forms. Okay, and basically RNA sequencing or RNA-seq approaches are very important here. Okay, and if you already know the genes or transcripts in a genome, okay, then basically microarray can be a quick method to know the expression level of those genes, right. So for example, for a pluripotent cell, like you can see that there are so many types of RNAs, okay, which are there, and it can be sequenced from like for example, somebody is interested in microRNAs, okay, regulatory RNAs or siRNAs, or alternate splice products for basically cancer and other tissue specific expression, or the mRNA is just for knowing that what all genes are expressing and as an evidence for the assembly part. Okay, so basically from a cell you can get, so this is only the mRNA and express RNA part, okay, which forms a product or a regulatory, but in addition there is a huge component from the structural RNA which are also there, and you have to do the ribodepletion steps to get rid of those structural RNAs. Okay, so basically RNA-seq is important with you can do this with low amount of total RNA extracted but high quality RNA. Okay, and basically it will tells a lot of the story about the functional aspects of the genome. And if you already know that what is the RNA, okay, the genome and its set of genes, then you can go for the microarray, and basically which can tell that the expression level of various genes, or even at the alternate splice forms of those genes, okay, when you are doing some time course or some comparative studies. Okay, so the strategy is like from the genome, from the cell, basically you extract the pre-mRNA, okay, and then fragment it. Okay, so basically this pre-mRNA gives the mature RNA which will be present in the cell, and you will be using that. This will give the RNA fragments, okay, and those RNA fragments, because RNA is unstable and the sequencing methodologies are more for the DNA, so you will convert that into the cDNA, okay, double-stranded cDNA, which will be sequenced by any appropriate sequencer. It could be long read or short read, or different type of library preparations are available, okay, and after that basically it will be aligned with the genome to tell that to identify the different splice variant, like this has 1, 2, 3 exons, it has just 2 exons which are connected to each other, and exon 2 is skipped. So this way it tells like the genes which are present, their respective positions in the genome, the total introns and exons in that genes, the various alternate splice products, and the relative expression level of each of those RNAs. Okay, so basically as I mentioned, so when you are doing this, you have to there are so many types of RNA which are present in the cell, like tRNA, ribosomal RNA, which are structural, or microRNA or other regulatory RNAs, and the mRNA which ultimately forms the protein product. Okay, and in prokaryotes it is easier to do because they do not have any exon intron structure, and determining these correct exon intron structure, the genic structure is easier, whereas in eukaryotes which have exon intron structure, it may be like more challenging. Okay, so basically in this part of our lecture we covered the different aspects in genomic and transcriptomic analysis that you should consider before going for a genome sequencing projects, and like what you have to do in that genome, like in terms of whether it's a genomic or transcriptomic or proteomic, and different or basically in the genome also what kind of information is present in terms of genes or repeats or heterozygosity or polyploidy. Okay, so these things you have to consider before going for a genome sequencing project, and basically in the next part we'll look at some of these things, how they have been applied to some genomes by using some case studies, and also we will discuss the concepts again while we are doing those case studies. Okay, thank you for today.